Browne from Western University up in London, Ontario in Canada. Now I'm going to be talking to you today about predictions and insights into bacterial sgRNA Cas9 activity with deep learning. So to start, when you're using CRISPR-Cas9, the goal is to cut DNA. And the way that this happens is that Cas9, the nuclease, is targeted to a section of DNA that is 20 nucleotides long with a single guide RNA that is complementary to that site, and with, of course, the adjacent PAM. In this case, we're looking at Streptococcus pyogenes, or SpCas9, and that PAM sequence is NGG. So there's a lot of two Gs beside each other, so there's a lot of potential target sites for SpCas9. Now, previously, work has shown that, you can predict these target sites using machine learning. Nucleotide sequences significantly impact activity. Now, what was noted was that although these predictions are great, there's potential for improvement by focusing on the data itself rather than the machine learning. Activity score readout is limited by control condition read counts. In measurement, what you have is sequencing for an experimental and a control condition. Both of these are read counts that measure cleavage activity of Cas9. And when there are not enough control condition read counts, activity can be impaired. Well, the readout itself can be impaired. Additionally, in terms of the model input itself, nucleotides outside the target site impact SpCas9 activity. Now I'll get into both of these things a bit more. But to show all of this, I'm going to be building three models with three different data sets. In purple, the Citrobacter rodentium TEV SpCas9 nuclease dataset. In green, the E. coli ESPCas9 dataset. And in blue, the E. coli wild-type SpCas9 dataset. These all range in size from about 30,000 to 60,000, which does go down a little bit after curation, but I'll get into that a little bit later. Overall, what I'm doing here is I'm building three different models, all with the same deep learning architecture called CRISPR-Hal. And then finally, I'm going to show the performance of the best performing model, CRISPR-Hal 2.0, on three independent training sets. So, to start, as I said, I'm not focusing on the machine learning side here. I'm focusing on how the data will impact performance. And to begin, what I looked at was how low control condition read counts can inhibit the dynamic range of Cas9 activity scores. So what we're measuring Cas9 with is sequencing readouts in both experimental and control conditions. And then we are creating a log fold change activity metric between the two conditions. However, when the control condition read counts are too low, what ends up happening is that you cannot successfully read out the activity in the experimental condition. So what I did was, with this value that I'm calling C here, I'm just using a cutoff, that C value, for a minimum control condition read count per target site, per sgRNA. And then I am calculating all of the subsequent log fold change scores and putting that through 5-fold cross-validation with each of the three models, each of the three training data sets. What this results in is a pretty fair amount of data removal. So for the wild-type Cas9 especially, the data set has about 50% of the data points removed, and that is the point at which the model performs the best. Basically, what can be obtained from this is that, even though it's a bit of a sin to remove so much data, these low quality scores are impairing model performance by having low dynamic range in terms of the actual measurable activity scores. Moving on from the scores, we then go to the actual model input itself, the target site sequence. And what I looked at was the impact of adjacent nucleotides, so things that were either downstream or upstream of the target site, And then using this same sort of cyclical process of, adding on one downstream nucleotide, performing five-fold cross-validation, and then extending by one more, and doing that also in the upstream direction. And what I found here was that model performance is pretty significantly impacted by these upstream and downstream nucleotides, especially for wild-type Cas9, where performance was increased by over 0.1 average Spearman correlation on the downstream side, and still also pretty strong on the upstream side, with less from TEV SpCas9 and ESpCas9 being kind of in the middle. And based upon this, we now have our curated scores and our extended input nucleotide sequences. So I then train up a final model for CRISPR-Hal 2.0, which is TEV SpCas9, as well as the ESpCas9 and wild-type SpCas9. And I show the performance here on their held out 20% test sets, which is pretty strong, all around the 0.7 plus Spearman correlation. Now that's great, but something that's probably more important is how well the data generalizes to a completely different data set measured in a different way. This is using these three independent test sets use an enrichment metric. They're in E. coli rather than CRISPR-Hal 2.0, which is Citrobacter rodentium. And, well, something nice to see is that the model does perform well here with upwards of 0.6 Spearman correlation on the two E. coli sets, and the Salmonella enterica set being about 0.74, which was awesome. In terms of comparisons, in purple here is CRISPR-Hal 2.0, and you can see that compared to even the wild type CRISPR-Hal SpCas9 created in this presentation, but also more importantly the prior models, including the original CRISPR-Hal, which is in yellow, this data curation improves performance across all tests. So overall, what we've seen here is that low read counts impair proper scoring, and that impacts model performance. Adjacent nucleotides do impact Cas9 activity, and their inclusion will benefit subsequent models. And that the data curation here does generalize. It's not specific just to the data set that you're curating. You're not focusing in on just one singular test set, but rather you are improving the data to result in an improved model overall.